So hello everyone, and welcome to today's session, Automating Workflows for TDM Analysis by LCMS. I would like to start by introducing today's speakers. Our first speaker is Dr. Mosé Barbaro. He's a biologist at the Ospedale Civile di Brescia. He has several years of experience in LCMS, including method development and system integration with LIS, as well as other laboratory environments. Mosé is in charge of managing the sector of pharmacology and toxicology in the hospital central laboratory. Our second speaker is Dr. Frank Kühlwein, who is the head of international sales at Chromsystems. He has a PhD in organic chemistry from the Ludwig Maximilian University of Munich. Frank joined Chromsystems in 1997 and is currently in charge of Chromsystems direct sales and distribution network. I will be the last speaker. My name is Gabriela Boza-Moran, and I am the Scientific Content Manager at Hamilton Robotics. I have a PhD in Molecular and Cell Biology from Royal Holloway, University of London, and an MBA in Management of Technologies from the Swiss Federal Institute of Technology in Lausanne, Switzerland. My role at Hamilton is to create and disseminate scientific content describing the use of Hamilton products in various application areas. Now, before we begin, I would like to mention some important aspects. First, this webinar is being recorded and it will be available for download later. We'd love to hear from you, so please write your questions in the box at the bottom. It's called Q&A. We will do our best to answer your questions at the end of the webinar. If for some reason we're unable to do so, please feel free to contact us after the webinar via the email. You will find this contact email at the end of the presentation. So now, the agenda for today's webinar is as follows. So Mosé will speak about the first 4 points that are related to LCMS basics, TDM basics, the hospital-specific workflows that they have in the hospital where he works, and the automation that they have set up. After this, Frank will talk about Chromsystems kits for TDM. And finally, at the end, I will present Hamilton Robotics automation solution for TDM. At the very end, we will have the Q&A session. So please, as I repeat again, write your questions in the Q&A box. And now I will hand it over to Mosé.
Good afternoon, everyone. Thank you for inviting me, for having the occasion to speak to you this afternoon. I will pass the first 2 aspects of the presentation just to give you the basic information about LCMS and TDM, and then we will focus on the main topic of this presentation. So what are the main steps of the workflow when we work on LCMS? Well, the first step, like in other methods, is to prepare the sample. We can just analyze the sample as it is. So we need a first step of sample preparation, which sometimes include enrichment, sometimes include extraction or other more complex process. Then we move to the chromatography part, and then with the analysis with the mass spectrometer that it's coupled with the chromatography part. And but for the last part, but not the least, the analysis, which consists in the evaluation of the data obtained from mass spectrometry. Okay, next one, Gabi. Can you move to the next one, please? Thank you. So now we are going to see a bit more in detail the 3 main aspects of an LCMS method. The first one is the mobile phase. Mainly, we can choose between 2 kind of chromatographic and mobile phase use. Which one is the isocratic method and binary gradient? These two methods have a main advantage, the second above the first one, because as you can see in the picture here on the right, the changing the isographic condition can change the time in which the peak exit from the column. But with a gradient, we can combine the advantage of both kind of isocratic method, obtaining a better resolution and best results. So this is an important aspect that someone who work with LSC need to know and need to develop for each method. Then we have to move to the chromatographic column, that it's a part of the chromatographic system. And here in the picture below the text, you can see a simple scheme of a chromatographic method, a chromatographic system with the AC pump, the column, and the detector. And once set up choose the column and set up the method, we move to the mass settings. The acquisition of the data from triple quadruple mass spectrometer, it's more complex compared with other detector. We have to consider that the acquisition in mass spectrometry is to be considered a three-dimensional acquisition. So we have intensity and retention time that it's common to all the chromatographic method, but we have also the mass to charge ratio. That means that we have a third dimension in our data, and this is the mass where the power came from mass spectrometry, because the third dimension allow us to have a more specific identification on what we are going to analyze. Okay, next one. Then we can use all these instruments, all the power we have seen from mass spectrometry and SPLC method to do the analysis for TDM. And what is TDM? Therapeutic Drug Monitoring. It's an important aspect in the therapeutic regimen for the patient. As you can see here in the picture, when someone take a drug, changing the dose can change the therapeutic effect we are expecting to obtain. Well, as everyone know, we have always desired effect when we take some drug and the toxic effect. The aim of the TDM is to personalize the concentration in each patient, to keep the concentration inside the desired effect, avoiding to go into the toxic effect, and to maintain the therapeutic effect above the inefficacy area that is below the desired effect. So we have a dose like 10. Our patient, we don't have desired effect, but neither toxic effect. If we are in the green area, the desired effect is maximized. If we go above and we go in the red area, the patient will have a for sure desired effect, but we have a very important toxic effect. And for some drug, the toxic effect can be irreversible. So we causing the damage to the patient that cannot be recovered after interrupting the drug. So this is important. This is why it's important to do the TDM. Next one. Just to give you an example of the TDM, I will not go in the detail about the drug which are referred to this work, but just to give you the idea of the four main questions which TDM answer. So why it is important to measure these compounds? In part, I already answered to this question, because we have to consider the plasma concentration in the patient, considering the regimen received. And this is considered in the whole population that can take this drug. So as you can see in the picture, the value for the patient is very low because it's the first 25 percentile. Then we can give a more specific question. So the plasma drug concentration in this patient is influenced by his medical condition. So we move from the general population to a population with a specific pathology that it's like our patient. And now we can see that for this kind of patient, the value is not so bad as it was before, because it's moving a bit higher, closer to the target area, that is the darker blue. Then we do a more specific question from this one. That is, how should the drug dosage be adjusted in order to reach the optimal circulation exposure target? So now we are analyzing in the specific data for our patient. So our patient, its target concentration is in this graph. So comparing with the target area, that is the green bar in the slide before, how our patient is going. In this case, it's not going well because it's below, part of its concentration, it's below the target area. So for some time during the day, it doesn't have the effect we are expecting for our drug. And then we move to the last question. So will TDM and subsequent dosage adjustment improve the patient therapeutic outcome? In this case, yes, because increasing the dosage, we can see that our patient keep staying in the blue area, so it has therapeutic effect with a little bit time outside. That means it can have some adverse effect, but maybe not so important, but we guarantee him or her that the concentration is always above the target required. Okay, next one. Okay, so just for example, for this session, we are using immunosuppressant as an example of TDM. So we can monitoring for TDM with TDM for immunosuppressant that are the main immunosuppressant used in transplant patients. That is grouped in calcineurin inhibitors that are cyclosporin and tacrolimus that, as you can see in the picture below, are acting on calcineurin pathway, blocking the IL-2 expression, and mTOR inhibitors that, on the other hand, go blocking the mTOR transcriptor factor that has the effect of reducing biosynthesis and promoting autophagy. That mTOR, if not blocked, do exactly the opposite as reported in the picture. Okay, next one. Now, just a brief example of what we do as hospital, in our hospital for TDM. We do with the SPLC coupled with classical detector, mainly dopamine and its metabolite, that you can see in the picture in the bottom corner of the whole pathway of dopamine. With the SPCMS, we do anti-HB, anti-epileptics, antimycotics, and immunosuppressant as the main topic of this presentation. And with the non-chromatographic method, we do the TDM for antineoplastic and antibiotics. Vitamin D is usually can be considered both an hormone and a monitoring for TDM because in some patients they are supplemented with vitamin D. So when someone is not supplemented, the vitamin D is just a normal hormone that we are going to monitor. But if it's supplemented, then we can consider it as TDM because vitamin D in highest dose can be toxic. Okay, next one. Now, this is the main topic of this presentation, and we are going to be a bit more detailed. So, automation. How is the sample flow? This is the classical flow in each laboratory hospital, and you can see that we start from patient. The clinician asks the test with the test ordering. we have a sample collection, the sample transport, a processing of the order, the examination of the sample, report the results interpretation, and back the results to the patient. In red, around this virtuous circle, you can see the probability to have an error. Obviously, the post-analytical, it's outside of our control because it's up to the clinician who made the order to interpret correctly the data. The pre-analytical part, it's in part of in charge of the laboratory because we can have some effect with our indication on the pre-analytical part, but not much. The most part where we can have effect, it's the ordering, processing, sample examination, and results report. You can click next, Gabby, please. Okay, this part in the green box is the part where we can have some influence. And this part, it's the part most importantly that we can automate when we work with LCMS. So we can automate the analytical part with the sample preparation, the worklist generation, and the result processing, and one of the post-analytical part that is the results release. So once the sample reach the laboratory, that is the last step of pre-analytical part, we can begin to automate, like for clinical chemistry laboratory where the automation is already in place. But we can do something even in our field where the automation is not so common. Okay, next one, please. Now we are going to talk about the Chromsystems and Hamilton Mass Star Microlab Star, that it's a CAVD device that it's present in our laboratory. And we are going to compare the difference of working with it compared with the manual preparation, because as you all know, the preparation for chromatographic method and chromatographic method coupled with mass spectrometry need a lot of work before we can put the sample on the system. The manual part consists in preparing the calibrators, preparing the controls, using the internal standard, the reagent, and the sample are the part that is common with both methods, because we can't avoid to use calibrator or internal standard or the reagent or the sample. But what change? It's what we need then. In our case, we need to use vials and the laboratory equipment to create the work list by hand, and operator with some skills and mainly the time for the operator. This part is covered by the Hamilton system. Okay, next one. Okay, so now we are going to overview a little bit more the difference between the manual process and the automated process. As you can see here in the left picture, we have the Hamilton for the preparation, and on the right picture, we have the Waters UPLC XIVO, that it's the LCMS method that we use, the instrument we use. In the middleware, in the cloud, there is the laboratory information system. That is our laboratory information system, and the middleware that allow everything to work. What does it mean? Okay, as you remember from 2 slides before, I said that we have the ordering request and the sample transportation to the laboratory. The ordering request came from the laboratory information system. This is the place where all the data are stored. Then this data move to our middleware. The middleware is the bridge between the preparation, the analysis, and the list. That it's the cloud behind everything. So we can put the main tube on the Hamilton system. Here we put the reagent, the calibrator, the controls, samples, plates, everything needed, and we start the CAVD method for the preparation. The system do the whole preparation, that in this case, the operator is free to do something else. And while the instrument works, we save at least 1 hour and a half of operator time. When the preparation is completed, we have the sample map saved on the Hamilton PC. Thanks to the middleware and the laboratory information system, this information go to the middleware. It's transformed by the middleware in something that the water system can read and program the instrument to do the analysis. This is important for the traceability of the sample, because the Hamilton read the barcode, it says where the sample is in the plate, and then this information is moved electronically directly to the analyzer. So no operator need to change anything going to enter the data, improving the quality of the process. And then the HPLC system can do its analysis. And once the analysis is completed, the operator has to do the report of the data. But once evaluated all the chromatographic information we have, this information can be moved back to the laboratory information system, always using the middleware. In this case, the middleware, we take the results, and considering that they are already associated with the barcode to the sample we have received, can be associated without any chance of error to the correct placement. So we are going to have a process from the sample, from the sampling of the sample. So when we start the preparation, to the report, to the final report, all connected with the digital data without the need of the operator to enter anything. And this, it's the next one, Gabi, please. The next one, please, Gabi. Okay, thank you. And this, one less, thank you. And this, it's the answer to this question. Which problem have been solved with automation? First, mainly the traceability from patient sample to the report. So, does the extracted sample correspond to the primary tube? This is, of course, because the Hamilton do the preparation, so use the primary tube and link the primary tube, the information with the barcode on the primary tube to the place on the map, on the plate map. So the file that Hamilton generate contain all the information to trace which primary tube was placed in which spot of the plate. Then the performed analysis correspond to the one that was requested. This is one of the most important aspect when the preparation is done by hand by the operator, because it's easy to put a sample in the wrong list and do one analysis when was requested another. With the automation and the integration of the system, this is not more possible, because we have a middleware in the middle that can do the barcode check and check what tests are requested inside the list. So if the one patient doesn't need to do one analysis, we have a check that can tell us that one sample doesn't need that kind of analysis. And so we can notice that and avoid this kind of error. And then another more important question, where the results send to the correct patients? Because if we have to enter the results by hand, we can just skip a line or enter twice the same results, and then all the patient, or a part of them, or just one, will receive the wrong results. That it's not acceptable in a laboratory, and we have to do everything to avoid this aspect. And this kind of problem can be resolved with automation. And where the results reported correctly, this is another important aspect, because if we are, even if we type the correct result to the current patient, just the point placed in the wrong place can change a 10 to a 100, and this is not acceptable anyway. And so the automation ensures full traceability from primary blood tube to the final patient report, that it's the same thing that we are used with the clinical chemistry and the high automation laboratory, moved to our section where automation is not so common, and everyone is used to work by hand. And the last but not least, the reduced hands-on time during the automation leave the operator free to do other more important activities, because operator that spend time to do the preparation cannot focus on the report of the results, to see the chromatographic peak and to correctly evaluate the results. But with the automation, we save time from monotonous and repetitive step to more important aspect in our job. So it always seems impossible until someone does it. And this is just a citation from Nelson Mandela. Now I leave the speak to Frank Kühlwein from Chromsystems.
Thank you, Mosé, for your transition. Hello, welcome to everyone. And Gabi, Federico, also my thank you for the invitation and the opportunity to speak at this event. I now have the pleasure to show you what commercial solutions for therapeutic drug monitoring on Hamilton Master platforms are available. But before I go into more detail, a brief foray and insight into our company and what we usually do in the TDM sector. Founded in 1990, Chromsystems have become a specialist in clinical diagnostics by HPLC and MS/MS. And yeah, since we started in 1990 with HPLC assays, the design and the manufacturing and production and marketing of HPLC assays, the time since then was very, very exciting. And I still remember that after the millennium, with the occurrence of mass spec in the clinical arena, it became for us a kind of evolution step and a breakthrough. Company went through a breakthrough, and where we were deciding to design and to produce and to manufacture also assays, ready-to-use assays for the tandem mass spectrometry. And in 2007, our first MS/MS assay was launched. This was exciting time, and 30 years have passed by now, and we are still growing. We are now over 160 employees. We have a pretty strong direct sales representative network in 10 European countries. We have exclusive distributors all over the world, and quality is our priority. This means also that quality, and now in light of CE IVDR in around the corner, it is in our focus, and company invests massively into this field in order to achieve and to cope with these challenges of a regulated clinical market. We have around dozens of people busy in the regulatory department and another dozen people busy in the QC and technical product management, just to achieve that standards and that quality. It's worth to mention also that we are constantly and regularly audited by TÜV Süd. Also, FDA has audited us without findings. We are complying fully to Canada Health, and this is also the reason why we are able to sell into the Canadian territory. We have also passed successfully Anvisa audit. That's why we have now also licensed to sell into Brazil market and Saudi Arabia market too. We are holding the license, and of course CE IVD, or that our kits are for diagnostic use, and CE IVD validated is a must-have and comes without mentioning. Next slide. Yeah, well, among many other important fields, such as newborn screening, vitamin profiling on one side, also the biogenic amines, we have, or I would say TDM, drug monitoring, is the product line, is the outstanding product line with a long history and tradition in-house. Next slide. Yeah. So there's a long history and tradition in-house for the therapeutic drug monitoring and building solutions, complete solutions around that. And because robotics and automation have more and more entered into the lab arena, clinical arena in regard to MS applications, we began to design and produce suitable, convenient, user-friendly turnkey solutions, combining validated assays with liquid handler like the Hamilton one, the software in one CE IVD workflow, featuring full sample tracking, clot detection, LIMS connectivity, and full walk-away operation. I like to call it a plug-and-play solution because also we take fully care of the startup implementation and the service, after sales service, and the support during the routine operation, during the routine run. And with Mastox immunosuppressants and Mascrom 25 hydroxyvitamin D2, we have validated protocols, and we were entering with these two ready-to-use assays. We were offering the first mass spec assays and solutions, or let's say we were among the top 5 that you have to have when it comes to clinical mass spec. Next slide, please. In the meantime, we have 9 turnkey solutions available. And those 9 turnkey solutions for Hamilton automation are integrated in our large TDM portfolio. Immunosuppressants, we heard before, presented by Mosé. We have, of course, also the mycophenolic acid available on Hamilton platforms, the anti-epileptic drugs, the catecholamines, free metanephrines, serotonins, the total metanephrines in urine, the vitamin D3, D2, the VMA HVA 5-HIAA in urine. And recently we added to that list the steroids and the vitamin B1, B6 in whole blood. Also as a contribution to keep our panel of products, our portfolio of products always a state of the art, and be able to provide solutions to the marketplace when there is a need and when there is a trend going in this direction. Next slide, please, Gabi. In this context, the Mastox TDM Series A, the simple concept of providing a one column, a one sample preparation, and allowing you the access to measure and to quantitate over 200 parameters. The drugs, inclusive also the metabolites, is an attractive one and has become a bestseller. With the results, ladies and gentlemen, that we have now available antiarrhythmics. We have answers for the measurement if you want to measure antidepressants, anti-epileptics. Let me speak also later about anti-HIV and anti-COVID-19 drugs. The antimycotics is very often asked, benzodiazepines, mycophenolic acid. The neuroleptics is very important because there is a lot of new neuroleptic drugs were released, and by the mean of providing an MS assay, we are much more faster able to offer a complete solution, a ready-to-use assay to the laboratories when there is a request for measuring of this drug, neuroleptic drug. Psychostimulants and TCAs are also available to round up this list. Next slide, please. It is necessary for a specialist like Chromsystems in this niche market to react and to respond quickly to new trends and challenges. And by launching an application to quantitate COVID-19 drugs in serum plasma by MSMS, we did so. And we are now featuring 9 drugs and metabolites in a 3.5 minute separation. We are providing multi-level plasma calibrator set, also the controls in 2 levels, plus a user-friendly workflow in order to allow you to measure and to catch up also and to provide to your doctors and we'll be able to tell them, we can report to you remdesivir, we can measure and quantitate to you favipiravir, the ritonavir, the chloroquine, and many more in this context. Next slide. And that's why I would like to say, that's why I would like to invite you to this journey. And there is a lot of new things in the pipeline, and there you can expect also a lot of new essays and new solutions from Chromsystems in this field, not only in the automatic field and robotic field, but also in the field of mass spec in general, clinical mass spec in general solutions. And I thank you very much for your attention, and I'm looking forward to your questions later on after Gabi's talk. Thank you.
Thank you, Frank, and thank you, Mosé. Just going to take over here. And I will go now to the last part. I would like to take the opportunity to introduce the diagnostic team, Hamilton Diagnostic Team, some of our products and modules for the automation of the sample preparation for LCMS, and also the content that we have developed for this particular topic. So an overview of Hamilton. Hamilton is a private group founded in 1947 by Clark Hamilton in a garage in California. Today, Hamilton has 2 headquarters, one in Reno, Nevada, and one in Bonadud, Switzerland, and a total of 5 manufacturing sites, which also include in Massachusetts, another one in Switzerland, and one in Romania. Altogether, the company employs more than 2,500 employees around the world, which include offices in 7 subsidiaries in Europe and China. Hamilton is divided in 5 independent business units. Hamilton Robotics, which is specialized in the manufacturing of liquid handling instruments, both for OEM and for direct sales. And this webinar is focused on the products and services for this particular business using. But we also have Hamilton Storage, which manufactures ultra-low temperature and automated sample management systems, as well as small devices for capping and decapping. Another business unit is Hamilton Process Analytics, which manufactures measurement devices, including sensors for pH, dissolved oxygens, and conductivity. Hamilton Laboratory Analytics, which manufactures syringes for use in various applications. And finally, Hamilton Medical, which manufactures ventilators for people in critical care. Before I introduce our diagnostic team, I would like to highlight the impact of a change that will soon take place in Europe. As we all know, Europe will dramatically increase the regulatory oversight of diagnostic products from 2022 onwards. We will go from the IVD directive to the IVD regulations. And this is expected to have a significant impact in the way clinical laboratories work in the future. This is particularly important for laboratories that are doing LC-MS analysis, given the fact that the majority develop and validate their LC-MS assays in what we call LDTs, or laboratory developed tests. Yet, from 2022, only LDTs for which an equivalent CE IVD test does not exist will be permitted for use. So in preparation for this, Hamilton has already developed 2 IVD compliant platforms, and I will talk about this in the next slide, and form a specialized diagnostic team to support our partners during this journey. So here is what we see on the right. This is a team that is led by Federico Sebastiani, who is also going to be present at the end of the webinar and available for questions. Notice this team is growing, and now we have sales specialists in China and other ones in the Nordics. Hamilton Robotics has 4 platforms for automatic liquid handling solutions. So we have the ML Prep, the Nimbus, the Star, and the Vantage.
Mhm.
And our line of IVD compliant platforms are currently based on the Star. We have developed 2 IVD compliant systems, so the MaStar and the clinical NGS Starlet, and 2 fully validated IVD systems, the Chromsystem MaStar and the Sentinat 200.
Mhm.
So IVD compliance systems, just to make the difference, are open platforms that can be used with the reagents of any kit provider. In order to become an IVD certified system, so we see on the right, the deck and the software package of the platform need to be locked to the workflows of a specific kit provider, and they need to be validated as such. And this is the case for the Chromsystem MaStar, which is locked to the kits for now. As Frank was saying, this is expanding. It's locked to the kits of Chromsystem and is validated for immunosuppressants and vitamin D3 and D2 and expanding.
Mhm.
The Chromsystem MaStar is a CE IVD certified solution. So our open platforms now can be equipped with various different tools and modules. And here I want to show some of the ones that are relevant for clinical chemistry or for LCMS. So first we have the flip tubes for close reactions. The flip tubes provide an alternative to plate serum automated assays. And furthermore, our platforms can be equipped with what we call the flip tube tool set that automates the opening and closing of the flip tubes. We also have the MP Square, which is monitor multi-flow positive pressure evaporative extraction. So MP Square sounds a little bit easier to pronounce. And this is for solid phase extraction with positive pressure. So we also have a vacuum-based SPE module for customers that prefer the solution. You can also equip with a centrifuge for spinning samples up to 4,200 RPM. So this is inside the deck. The ODTC, which is the on-deck thermal cycle for incubations between 4 degrees to 99 degrees. The punching module for card punching. It recognizes positively stained samples and negatively stained samples, and it can make punches of 1.2, 2, 3, and 6 millimeters directly into the collection plates. The APH module for pH measurement. The sealer for adhesive plate sealing with optical, clear, or pierceable foil. We have some Hamilton specialized tips for accessing samples in various seal labware. And the last 2 tools are based on the partnership with other companies. So first we have specialized tips, and here we have partnered with 3 companies, so IMS, DPX, and Finexus. And we have automated their in-tip sample purification. With some tips, samples can be filtered, and with other samples undergo in-tip chromatography. So they bind to common affinity resins. Finally, we also partner with Cyanova for the automation of their express dialysis solution, which permits to do on-plate buffer exchange. So I would like to invite you to take a look at the content that we have recently developed around this topic. We have an ebook which talks about TDM analysis, why automation is important in this area, and what we as Hamilton can offer. We also have a case study with Mosé, where he explains in more detail the automation of immunosuppressants in the Chromsystem MaStar, and this webinar, which will be available for those who could not, well, for everyone, but also for those who couldn't participate today. With this, I would like to end the presentation and open the floor for questions. I will now be moderating the Q&A session, and Federico will join to answer questions related to Hamilton's IVD platforms. See. Okay. It's coming, okay. So one question is it the Hamilton master unable also for non-IVD applications? So can we use a master also for non-IVD applications? Maybe Federico, you want to answer this question?
Yes, good afternoon to everybody. Yes, of course, the master, let's say the Chromsystems, if you refer to Chromsystem master IVD compliant, can be used for non-IVD applications. The thing that has to be taken in consideration is that is not, let's say, we have to protect our program and the environment. So we can suggest to use, for example, a second PC to connect with the system in order to use a research use only environment in the PC, and this will be the the easier and also more safe way to use the master IVD in also for non-IVD applications.
Thank you. Another question is, are there Hamilton distributors in Mexico? I think it's also for you, Federico.
Yes, yes, of course we have distributors in Mexico from Hamilton. Just one point is that here we are talking about as Hamilton employees for the part of the globe that is reporting to Bonaduce. So Hamilton, as Gabi showed in the previous slides, has two headquarters, one in Reno and one in Bonaduce, Switzerland. So Mexico is part of the Americas, of course, and so it's managed by the Reno territory, by US, let's say. And at the moment, the master is not sold in the US territory.
Thank you. Another question is, to which MS supplier can you link your platform? I'm afraid that this is again for you, Federico.
Yeah, to any supplier. So there are no limitations. So we are talking about it to all the principal supplier of a mass spectrometer.
Yep.
Okay.
Same for the kits. We do not depend on a particular MSMS brand. So we are guaranteed that our kit work on any MSMS platform and MSMS system. And that's why we at Chromsystems have almost all MSMS systems that you can see in a clinical arena. We have also them in-house in order to be able to give this guarantee and to ensure that the customer, what any platform he has got, MSMS platform, triple quad, that we can guarantee that we can service support the kit from the first day when the kit arrives.
Okay, we have two more questions here.
Mm-hmm.
Can the open IVD systems handle magnetic beads for sample preparation, specifically wash and dry steps?
Federico.
Federico.
Yes, I would say that the IVD systems is not prepared, let's say, in this way. But what we can offer is the open platform that can manage also the magnetic beads. but the IVD system, the Chromsystems IVD system, is not prepared to manage magnetic beads.
Correct.
Another question here is, are you developing special interface for each supplier?
If you mean from the supplier, I imagine that the supplier is the, let's say, is the kit provider.
Mhm.
So in that case, special interface, I can interpret that is the method, because in our world in automation, we are talking about a method. So yes, it's possible to develop the method for any supplier, but at the moment we have the Chromsystems. The master IVD is for Chromsystems. It is branded Chromsystems for this reason.
Right. And we develop in Chromsystems R&D department. We develop this. So we have Hamilton platforms, of course, and that's where we are developing the scripts for a certain assay. So this is part of the solution of the turnkey solution. Is the script. This is also part of the IP of the know-how that we are now integrating in this kit, in this solution. And that's the work or the time and the labor the customer finally does not have to do, because this job is done at Chromsystems, and that's part of the solution we provide, and make it become a plug and play solution.
There was a follow-up question about this. Are you developing a special interface to MS supplier to transfer data?
Yes, it's not really a special interface, but as I said, the master Chromsystems is able to transfer the, let's say, the data to connect with any LCMS system. So it's just a matter to exchange the data, and this is done for any principal provider of LCMS systems.
Yeah.
I have one last question here. I think we are getting to the 1 hour threshold. The question is LCMS part also included in one CE-IVD workflow? If so, which MS supplier's instrument is validated?
Okay. Federico, okay if I answer?
Yes.
Yeah, like I said before, we do not stick to a particular brand or specific platform. Again, from the most current MS platforms that you see in the clinical arena, in the clinical laboratories, we have those platforms in-house, and those are, and in a design and development process of an assay, either on our robotic platform, but also if it is a manual workflow, we are of course have to ensure that we validate our kit on a specific platform. And this is specifically from that can be specifically from platform to platform different. So on any platform, SciX, Waters, and so on, we have of course also to validate specifically our assays, our kits on that. And that's why it is it takes resources, it takes know-how, it is we have a large team for that, but it is also at the end a solution, or it's part of the solution we give to the customer, that this is a fully validated method, and we can guarantee that we can set it up and get it up and running on a platform X, Y, Z.
Okay, and I think that was our last question. We are just getting to 5. I want to thank all the speakers and everybody who attended. I know there was a problem with the people who register at the last moment. We will send this the recording of this webinar later. And yeah, so thank you very much and have a good afternoon.
Thank you all. Have a good afternoon.
Thank you to everybody.
Thank you. Likewise. Thank you to all. Goodbye.
Bye.
Bye.
Goodbye.